Welcome everyone to our Cancer Genomics Cloud webinar for May. I am really excited to kick off this two-part mini symposium on dogs and cancer. We have three phenomenal speakers this afternoon who will be speaking about the genomic and computational resources available for canine cancer. And so I want to get to them right away, but first, a little bit of housekeeping. I would like everyone to please type your questions into the meeting chat. And I will voice your questions for the speakers at the end. This will just help us have a smooth show. So without further ado, I want to go ahead and present our first speaker, Dr. Cheryl London. And let me just get things set up so that Dr. London, you're welcome to start sharing your screen. Dr. Cheryl London is the Anne Engen and Dusty Professor of Comparative Oncology at Tufts Cummings School of Veterinary Medicine. She's a seasoned veterinary medical oncologist with a focus on translational and comparative oncology. And as Associate Dean for Research and Graduate Education at Tufts, she has expanded the university's research footprint and increased DVM student engagement in research activities. Dr. London earned her DVM from Cummings School and completed her medical oncology residency at the University of Wisconsin-Madison and received her PhD in immunology from Harvard University. So take it away, Dr. London.
Hi, so thank you very much for inviting me to share with you some of the advances that we have achieved in the canine genomics toolbox. And I just want to mention that Gina Kuffel is going to take over at the very end of my presentation to talk about a really unique resource, the Integrated Canine Data Commons. So for those of you that are not aware of it, I think it's really become clear to all of us in the canine community that cancer is a leading cause of death in our patient population, and therefore potentially represents this enormously beneficial translational model for us to understand cancer genomics and also for us to understand and develop novel therapeutic approaches. So if you look at causes of death in young dogs and juvenile dogs, overwhelmingly it's infectious disease, traumatic disease, et cetera, with neoplastic disease very, very low. But when you get to the adult population, and usually this starts around age 5 and then peaks somewhere around 10, 11, and then starts to fall off. Neoplastic processes are the major cause of death, which is really a little bit different than the human side, where cardiovascular disease represents a really large component of human death. So dogs predominantly die of cancer, and the yearly cancer incidence is actually quite high, and it's estimated to be much, much higher than it is per 100,000 dogs than it is per 100,000 people, almost 5 times greater than in people. So dogs get a lot of cancer and they die of cancer. And, so for many, many years, really actually for 3 decades, there's been a real growing interest in leveraging this population of dogs with spontaneous cancer to better understand how cancer develops and to potentially explore novel therapeutic approaches. But there have been historical barriers that have really impeded progress. And some of those included really the lack of knowledge regarding canine tumor genomics. Like we just really didn't understand where they overlap with the human cancers as well as mouse models. We had incomplete canine genome builds with inadequate annotation, and we had few tools to interrogate and analyze genomics data. And then lastly, we really had poor connectivity with other genomic resources like human mirroring and other data sets to facilitate comparative studies. And this really was discussed in depth at a meeting that was held in the National Academies in 2015, where the communities of human and veterinary and pharma and everybody really involved in cancer research came together to discuss these barriers and to chart a path forward. So just in general, much of the progress we've made has really been in the last 9 to 10 years. So one of the big things that's more recently that's happened is really the generation of a much better canine genome build, CanFam4. And this was recently published, and I think it addressed some critical issues that we had with the original build of the canine genome, which was good, but had many gaps in it that really made it difficult for us to essentially carry information that we were generating in dogs over to the human side and back. So CanFam4 has chromosome length scaffolds and contiguity increases. That's 55-fold over CanFam 3.1. And it actually includes a lot of really important elements that were absent, including previously hidden functional elements, including promoters, other genes that were missing actually from the genome build, microRNAs, and lots of non-coding elements that are turning out to be really important. Other key genomic regions were completed, including the DLA, T cell receptor locus, and 366 cosmic cancer genes. So what really happened is that it filled in a lot of gaps. It really addressed a lot of the architecture issues that existed with CAN-FAM3 and provided us with a template that is far better moving forward when we think about analyzing the data sets that we're getting from our canine cancers that are being looked at right now. So just to give you a sort of a deeper dive into this, if you look at the gap regions, there's now a large number that are now contiguous in CAN-FAM4. There are over 7,000 regions that contain exon regulatory elements that are now included. So it's a much more accurate gene model. There's improvement of functional areas that we just mentioned, and actually 8 new genes were found and included. But I also want to point out one of the really big advantages of having this new build, which is the elements in the non-coding region. We know that there are large elements of regulatory regions that are involved in disease. And it's becoming more and more clear that these regions that are outside of coding regions are really critical for cancer and other disease regulation. And one of the big things that's happened over the past couple of years is an effort to map those conserved non-coding regions across mammals. So basically, if you can find all of those conserved coding regions across a huge number of mammal genomes, then you can basically conclude that those are critical elements because they're conserved across species. And those were absent, really, or in large part absent from the original build. So now we have those conserved non-coding regions in the dog mapped, and those are functional. And we're learning more about how they're contributing to disease processes like cancer. In the second part of this webinar, Dr. Elaine Ostrander is going to talk a bit about the International Dog 10K project, which is another huge effort that is going to add, or has added, substantial value to the breadth of understanding that we have about canine genomics through its sequencing and analysis of several thousand canine genomes. So what this has enabled us to do with all of this new sort of genomic information is to really begin to understand where we see cancers in dogs overlap with our human counterparts and where there are opportunities for us to begin to use these models much more effectively. So the table that I have here just shows some of the genes that we know are mutated in specific cancers in dogs and where they're found similarly in humans. So in canine osteosarcoma, we see a mutation in P53, P10, DMD, SETD2, copy number variations in MYC. And you can look across human cancers and see a similar set of genomic alterations. The same is for B-cell lymphomas, hemangiosarcoma, urothelial carcinomas, and mast cell cancer, which is actually a cancer that is not common in people, but very common in dogs, and in dogs it carries a KIT mutation. And so KIT mutations in dog mast cell cancer really act as a model of KIT mutation in human cancers, and a human cancer that has a lot of KIT mutation is called GIST. So what we're learning is that even though the histologies don't always match up, in some of the cancers, we can find matching genomic alterations. So we can study how these genomic alterations are different across different kinds of cancers by using this comparative approach. And just in another set of data that's been generated by Dr. Eleanor Carlson's lab, they looked at a broad swath of published data that's now available regarding the genomic landscape of canine cancers and asked if they overlap with what's known about mutations in human cancers. And if you just look at all mutations, you can see that the overlap isn't as great as you would like it to be. But when you look at driver mutations, when you just select for those mutations that are known to be consequential in the context of cancers, there's over 50% of them that overlap with human cancers. And so that really does suggest that there is significant synergy between these two diseases or cancers across human and dogs, and that there's a real opportunity to understand the driver genes and how they function in the context of these cancers across humans and dogs. Another thing that we've been able to do with the new genome builds is to really interrogate the structural components of the cancers that we see in dogs. And one of the cancers that we study quite a bit in dogs is osteosarcoma, which is similar biologically to the human counterpart, osteosarcoma, which occurs primarily in kids and adolescents, and occurs much more frequently in dogs than it does in the adolescent population. And it's been known for a long time that the osteosarcoma genome in the human population is structurally complex. And so with the new genome build, we were able to really do a deep dive and ask, is the genomic landscape of canine osteosarcoma similar to that of human osteosarcoma? And therefore, would it be a good model to study the genomics of this disease in dogs and potentially identify novel targets for therapeutic intervention? And the answer is yes. And so what I've shown you on the slide are two circuits plots, one for humans and one for canines. And you can see that there's very complex somatic rearrangements, localized hypermutation, frequent copy number aberrations, and relatively few point mutations. And so these are shared features across the disease. And again, having the genome builds that we have has really enabled this to be analyzed much more efficiently. Lastly, I just want to mention another element that has begun to be used quite frequently in our patient population, which is the liquid biopsy. And we have been able to do this because we have new genomics tools now. And liquid biopsy really represents a process by which you are sampling the peripheral blood, essentially, for elements of tumor that are released into the bloodstream. And this is typically circulating tumor DNA, but it can be RNA, it can be protein, et cetera. And liquid biopsy has really become a surrogate biomarker for the tumor itself. And because we now have a really good template to analyze the genomic landscape of canine cancers, we can then really dive deep into the liquid biopsy world in dogs and begin to leverage canine cancers to study, improve, optimize platforms for improving liquid biopsy detection. And this here just is an example of one of our studies where we were able to show in dogs with osteosarcoma that they actually have high levels of circulating tumor DNA at baseline. And then when their tumor is removed by amputation, you can see that that drops down to 0, and that you can actually detect the circulating tumor DNA by ultra low pass whole genome sequencing 1 month prior to actually seeing clinical evidence of metastatic disease. And so this just represents the actual liquid biopsy readout here, and this represents remission, and this represents recurrence of disease. So again, really new powerful tools that have enabled us to do really detailed work characterizing canine cancers. Beyond some of the basics, which include just creating a really good canine genome build, has been the input from essentially biotech pharma in terms of supporting some of our efforts to create tools. So we now have a number of partnerships that are working to enhance our genomic toolbox. The first one that I just wanted to speak about was with NanoString, where we worked together to build 2 different panels that were canine specific, one for their nCounter platform and one to enable spatial transcriptomics on their genomics platform. The nCounter platform allows us to interrogate 800 canine genes for pan-cancer immune response studies. So this is the canine IO panel. It really sort of aligns very nicely with a human IO panel that's currently in use in human clinical trials. And it really has significant overlapping content that's designed essentially for us to do these direct comparisons. The companion is the Canine Cancer Atlas, which again was designed specifically to interrogate a number of canine immune responses to IO treatments. This one actually has almost 2,000 genes. Tumor-specific content for several cancers, including melanoma, osteosarcoma, lymphoma, bladder cancer, and glioblastoma, and again has overlapping content with the canine IO panel. So these 2 platforms are actually complementary. And I'm just going to show you some data that we've generated and published from using the canine IO panel. And in fact, it's now being used quite widely. In this set of data, I just want to mention that we had a clinical trial in dogs with diffuse large B-cell lymphoma, and we were able to, using the canine IO panel, identify genes that were associated in the baseline tumor with poor versus good response. And so this is sort of the output read. across all of our cohorts. But then when we did a deep dive into specific cohorts in this clinical trial, again, you can find the ability to discern those that did well versus didn't do well in this specific cohort using the canine IO panel, and then confirm differences in gene expression that we identified in the IO panel using real-time PCR. So this has been a really useful tool. In particular for this immuno-oncology study, it was able to identify which patients would likely benefit from this specific immunotherapy. In another example of using the IO panel, we were able to look at primary osteosarcoma tumors and identify which genes were associated with good versus poor survival after treatment. And we identified some key genes, CXCL8 and IL-6, which were associated with a worse outcome. And actually this confirmed prior published data, and then upregulation of genes associated with an innate immune response that were associated with a good outcome. So again, the IO panel being incredibly useful for us to identify novel biomarkers. In an example of how we've been able to use the canine spatial transcriptomics panel, this canine cancer atlas, this is an example of canine osteosarcoma tissue slide where we've stained for macrophages, T cells, and tumor cells. And we were able to look at regional differences in macrophage gene expression. And we were able to show that there were actually mixed gene expression profiles. So there was no predominant gene expression profile in the macrophages, although they tended to segregate in neighborhoods, essentially. You could get neighborhoods that had mixed, neighborhoods that had more M1 versus M2 or M2 versus M1. And this is really concordant with similar data from humans demonstrating these sort of regional differences in macrophage phenotypes across the microenvironment. And then we were able to look at T cell rich versus T cell poor regions and identify cold tumor sites with limited T cell infiltration and hot tumor sites with very high T cell infiltration. Again, pointing out the heterogeneity that's inherent in these canine cancers and really exemplifies why they sometimes can represent a real a model that truly recapitulates the human cancer landscape, where it's not clear that there's any one exact sort of neighborhood that dominates. In many cases, there are different neighborhoods, which represents the challenge with immunotherapy, which is that some regions are amenable to immunotherapy and others may not be. Another partnership that we've developed really over the past year is with 10x Genomics. They have several different platforms, including one for single-cell RNA sequencing and another one for spatial transcriptomics, which uses a slightly different platform than the genomics. Historically, these two platforms have been limited to using live single cells and frozen tissues due to the lack of canine-specific reagents. We recently partnered with them to build a canine-specific panel that enables us to use formalin-fixed specimens for both platforms, which really is going to open up a huge window of opportunity for us to go back to archived samples and do single-cell nuclei sequencing from all of those paraffin-embedded specimens. So there's just a huge, rich resource of unexplored tissues that we now have available to us. So this is all in the process of being validated, and hopefully these panels will be available next year. Just to give you a sense of how single-cell RNA-seq has really been able to help us define what we're working with in canine cancer, and particularly in the field of immuno-oncology. This data was recently published by Steve Dow's group, where they actually looked at peripheral blood mononuclear cells in dogs and found 42 unique populations by single-cell sequencing. And this is just an example of what this looks like. So this really, again, exemplifies how when you have the ability to dive deep and understand the genomic landscape of what you're working with, you can find all of those individual cell types and populations that really mirror what we see in humans. And in this set of data over here, we were able to begin to explore what happens after you treat a dog with lymphoma with an immunotherapy within the first 2 hours. And so this is the baseline single-cell sequencing here. And you can see this sort of rapid shift within 2 hours of treatment with an immunotherapy that really translates to this sort of loss of immature B cells, changes in translation and nucleosome assembly gene set. So this sort of data mining is becoming really important for us to understand biologically what's happening in a patient after they're treated. And again, really defines how we can use some of these canine cancers to begin to really deeply understand the changes that occur when we use novel therapeutic combinations. The two last things I want to mention before we go into the ICDC are the resources for comparative studies that are now available. The Comparative Oncology program has been around for probably almost 2 decades now, and that is embedded in the National Cancer Institute and supports a number of efforts, including a Comparative Oncology Trials Consortium, a Comparative Brain Tumor Consortium, and it's linked very closely to our more recent pre-medical cancer immunotherapy network for canine trials, which was funded by the Cancer Moonshot. And this is a large collaborative that's supporting this translational work that is generating a lot of the data, particularly the genomics data in canine cancer. And lastly, the CTSA One Health Alliance, which is really an organization of veterinary medical schools that have partnered with their human medical centers under this CTSA award to really jointly try to advance translational science. And this has generated a number of tools that are now feeding into the cancer work, including a smart iEquip platform, electronic health record transformation so we can connect all of our data sets together, and then a number of training modules that allow us to effectively, efficiently, and appropriately engage in clinical trial work and collect all the samples that are used for our canine cancer genomics work. And with that, I will turn it over to Gina to talk about the Integrated Canine Data Commons, which really is the linchpin of all of this effort.
Thank you so much, Dr. London. I want to briefly introduce Gina Kuffel. Gina is the technical project manager and product owner for the Integrated Canine Data Commons and works at the Frederick National Laboratory. And Gina's professional interests include bioinformatics, open source tools, cloud computing, and building web applications. And I am very excited for Gina to tell us all about the new integration with the Integrated Canine Data Commons and the Cancer Genomics Cloud. You get my...
Hi, Sarah. Thank you. Can you hear me? Okay. And can you see my screen, the appropriate one?
Yes.
Okay, great. Right. So I'm here to introduce the Integrated Canine Data Commons. So the NCI Division of Cancer Treatment and Diagnosis, DCTD, tasked the Frederick National Laboratory for Cancer Research with building the Integrated Canine Data Commons, which is a cloud-based repository of spontaneously arising canine cancer data. So not only does the ICDC provide a home for the data sets that are generated, but also aims to lower the barrier to building cohorts and then going and moving forward to conducting a meaningful analysis. So I prepared a very short video to demonstrate the utility of our current data portal. And actually, I hope that I enabled the sound, so please let me know right away if you can hear it.
I'm going to give a quick overview of the ICDC web application. So we're taking a look at the data portal here, and this is the home page where you can get some basic information, including information about submitting data, our programs, about the ICDC mission, and the ICDC news page. On the news page, you can see featured videos, an image gallery, some upcoming announcements, and also a featured tweet. We have a studies page where you can get information about each of the studies that is present within the ICDC. Right now, we are grabbing some of the information about these studies from live application programming interfaces or APIs, so that is why the load time for this page. So here we feature a data availability landscape tool where you can look across studies and understand if there are case files, study files, image collections, publications, or additional data within CRDC nodes for each of the studies in the ICDC. From here, you can click into any particular study to get more information, including information about the arms and cohorts of the study files that are available, publications, and supporting data. We also have a histogram that will show you the profiles of the sample site, sample type, and sample pathology, and information about the case file types, and more granular information about the PIs and the publications. We also have our data model navigator for exploring the ICDC data model. You can click into any of the nodes, so it's an interactive experience. You can also use the faceted filters to filter this data model down. And there are two views, including a graph view, which is the default, and also a table view where you can inspect properties. You can get templates for data submissions, or you can download a guide to aid with that data submission. We have some static pages that allow you to query our database, and also some information about the ICDC steering committee and working groups. But here today, we will focus on building a cohort. This is using the ICDC Explore dashboard page. You can see we have a series of widgets that will give us information about the programs and studies, the breed, diagnosis, stage of disease, sex, and file types associated with the data. So if we look, we have a pretty comprehensive faceted filtering collection. So here we can toggle open each of the facets. In this particular case, I'm going to open the study facet, and I'm going to select the glioma 01 study. And as I scroll down, I also want to open up the breed facet, and I am going to select bulldogs, which I could see that there are 4 cases that match my criteria. You could see that the widgets will update as I make my selections, as will the dynamic query bar interactively build a query at the top here. So maybe I'm interested in hemispheric as the sample site, and for the file types, I am interested in whole genome sequencing files. So I see there are 2 cases that match my criteria, and that will give me 2 case files. So I could see these are BAM files. I think I also would like to include the BAM index files for these cases. And now that I'm happy with the cohort that I've built, I'm going to add files for all of the cases that match my criteria. So I could see that 8 files were successfully added to the My Files cart page. And I could see within this page that all 8 files indeed are in my cart. So there are a few things that I can do on this page. I can read more about the functionality of the concept of a file manifest within the CRDC. I can learn about importing the ICDC file manifest to the CGC. And I could also learn about the new export to the Cancer Genomics Cloud button. And there is a tutorial that I can visit if I want to learn more about this. So from here, I can download the file manifest to my local computer. We take a look at that. I could see that we get file names, we get DIRS URIs for each of the files, and some amount of metadata for each of the files. So I could at this point take this downloaded file manifest, and I could go to the CGC, and I can use the import DIRS manifest function to get those files available within the CGC. But alternatively, we have a new single-click button that I would like to showcase. So here, if we open up the available export options, we see there is a new Export to Cancer Genomics Cloud button. So I'm going to click on that. I'm going to log in with my ERA Commons account. And this is actually going to put me to a multi-factor authentication scenario. I'm going to go ahead and authorize the Data Commons Framework service to use my information. And now I can go ahead and I see that this is the CGC interface, and I need to select a destination project where I will export my files. So I've set up a project called ICDC Export Button Demo. Click on the disclaimer here that I understand, and I'm going to click on Import Data. Here we could say the import started successfully, and this is telling me I could follow progress in their navigation bar. So up here in the Activity Center, I could see that indeed these files have began to import from my DERS manifest that I have provided. So this data is being streamed from the ICDC bucket and using that DERS compliant file manifest created through the ICDC. So I could see that all 8 files, including one additional file, which is the actual file manifest, have made it over to the CGC into my project. So if I open up that file manifest, I could see this is going to look very similar to the file manifest that we were able to download locally. It is not the same file, but it contains the same content. So I could see that I have my whole genome sequence files and all my BAM index files. And these are all the bulldogs that I was interested in and metadata for those files. So from here, I could head back to the files page. And now that I have my BAM files, I can select from over 900 public applications. There are workflows and tools and also interactive web apps. And I have the capability within the CGC to build my own workflows. So I will stop there. Thank you so much. And I think we're taking questions at the end.
Thank you so much, Gina. That was great. Yeah, so we will go ahead and take questions at the end. I'm going to introduce our last speaker now. It is my great pleasure to introduce Dr. Elaine Ostrander. She is the Chief and Distinguished Senior Investigator and Head of the Section on Comparative Genetics at the National Human Genome Research Institute at the NIH. Dr. Ostrander's laboratory studies the canine genome and its utility as a system for informing human health and biology. Underlying her work are extensive worldwide studies of canine population structure. She's published over 400 papers and reviews, won several awards, and in 2019 was elected to the National Academy of Sciences. Dr. Ostrander's current work focuses on the identification of cancer susceptibility genes in dogs, particularly bladder cancer and histiocytic sarcoma, the role of genetic variants in canine behaviors, the origins of dog breeds, and genetic studies on dogs from Chernobyl. So without further ado, please take it away, Dr. Ostrander. Oh, you're muted. No, you're still muted.
Okay.
There you go.
All right. There
Gotcha.
you go. I got all the right buttons. So thank you very much for those kind words. And it's a real privilege to follow the previous 2 speakers. And I'll try and do justice to their spectacular work. So I am going to talk about invasive urothelial carcinoma in the Shetland sheepdog, but I'm going to be talking in the context of the germline. We're going to focus exclusively on the germline and not on the tumor, and also talk broadly about approaches as well as resources, particularly Dog10K. So just by way of background, there's a lot of dogs in the US, almost 90 million, and that makes them as a population or as a species very amenable to genetic studies. They live in and they're exposed to the same environment as humans. They get the same diseases as humans, and they receive extensive preventative and diagnostic health care. In 2022, you may not know, but Americans spent about $137 billion on their pets, and about half of that was on healthcare. So all these things in aggregate get us really excited about understanding the genetics of cancer susceptibility in dogs as well as in humans. In dogs, we can sample very large populations of unrelated dogs in order to do genome-wide association studies. And we'll talk about so-called GWAS in just a few moments. We can also sample multiple generations of a large family due to the dog's relatively short lifespan, which is about 1/8 that of humans. So my lab works in both human genetics, human cancer genetics, and canine cancer genetics. And when I think about our human studies, they're expensive, especially doing sample collection. Families are small, 2 generations is generally what we have. There's lots of genes responsible for any phenotype or any trait. And the same genotype can often produce a lot of variation in the phenotypes. And that's because genetic background is complex, right? There's lots of different backgrounds that have come to bear when we think about even a region like the United States today. And as people, environmental exposures for humans are varied and they're certainly complex. Whereas in dogs, we have some advantages, and they all relate to breed population structure. In aggregate, they really increase our power. Overall, there are fewer genes controlling each trait. The genetic background effects are quite a bit less. We can identify gene interactions, and the exposures are more uniform. And I put this picture of long-haired dachshunds here just to remind you how members of a breed are so similar one to another, much more so than, for instance, members of a family are. So these are my current favorite breeds. And what's really striking about this, of course, is how similar members of a breed are one to another. And the important thing to keep in mind is that's not only at the trait or the phenotypic or aesthetic level, but that's true at the DNA level as well. But when we look across breeds, when we consider the Dalmatians versus the Basset Hounds versus the Bernese Mountain Dogs, there's a lot more variation in their appearance, in their lifespan, and so many things about them. And in their genomes, as we compare across, there's a great deal more heterogeneity as well. So we take advantage of all of those things in order to do trait mapping. Just a couple reminders: all dogs are members of the same species, Canis lupus familiaris, despite all that breed variation. What you may not know, though, is that most breeds are very recent. Most have only been around for about 200 years, and that offers us tremendous advantages in studying the susceptibility of any trait. My lab is in a long-term partnership with dog owners, dog breeders, dog lovers, dog veterinarians. We collect and we store DNA from all breeds, as well as from non-breed populations. We're not clinicians. We don't breed dogs. We don't keep dogs. We don't kennel dogs. This is a citizen science project. To date, my lab has over 45,000 dog DNA samples that we have collected and stored. All right, so everything we do goes back to this single figure. And this particular version is from 2017. It's now much bigger, much more complex. But for simplicity, I'm showing you this one. This is a neighbor joining dendrogram, where we're looking at how dog breeds relate one to another. So in this particular example, Heidi Parker in the lab took DNA from several thousand dogs, representing 161 dog breeds, about 10 dogs from each breed, and interrogated them at 170,000 random positions in the genome. And then she asked computer programs, okay, how do these organize? I mean, who's related to who, what to what? And you can see that those 161 breeds divide themselves into about 23 different clades, and they're indicated nicely by the color coding. So over here are the bully dogs, like the boxer, the bulldog, the dog de Bordeaux, the French bulldog, et cetera, et cetera. Up here we have a whole bunch of the terriers. Down here in red we have the spaniels, followed closely by the retrievers. So this is important because this tells us how to structure our genetic mapping studies. Now, as you certainly gleaned from the previous speakers, many breeds have an elevated risk for specific cancers. So osteosarcoma we see in the long-lived breeds, bladder cancer, which I'll talk more about, Scottish and West Highland White Terriers, as well as Shetland sheepdogs, histiocytic sarcoma in the Bernese Mountain Dogs and the flat-coated retrievers, and gastric cancer in the Belgian breeds, as well as in the Chow Chow. Now, I focus on invasive urothelial cancer as part of a long time and incredibly productive partnership with Debbie Knapp, who is the clinician here, as well as so many other talents. I'll just remind you, if you don't know, that IUC is the most common cancer of the urinary bladder in dogs, with about 40,000 new cases a year. The tumor develops from the transitional epithelial cells that line the bladder, and it invades deeper layers of the bladder, including the muscle wall. It can spread to other organs. Dogs come in with difficulty urinating, blood in the urine, or lameness due to the spread of the cancer. Importantly, this is a naturally occurring cancer. It closely mimics human high-grade IUC in terms of age of onset, pathology, cellular and molecular features, metastatic profiles, and treatment response, much as you heard about osteosarcoma previously. About half of affected dogs will experience metastases to distant organs, and the median survival time is very poor, typically less than a year. Dogs receiving multiple sequential protocols may live beyond a year. And we know that, as with humans, there are environmental factors that can reportedly increase risk, such as smoke. About 80% of canine IUC tumors carry a somatic BRAF mutation. So this is the one thing I'll say about the tumor and not about the germline. But it turns out to be important as we talk about genetic susceptibility, because it increases activity of the MAP kinase signaling pathway. And that's something that we see in humans, the relevance of the MAP kinase pathway as well. All right, so these are the top breeds for getting invasive urothelial cancer. And you see topping the list is a Scottish Terrier. So if you're a Scottish Terrier, your odds of getting the disease are about 21-fold higher than the average mixed breed dog walking down their street, followed by the American Eskimo dog at 6.5-fold, the Shetland sheepdog, the West Highland White Terrier at about 5.8-fold, and so on as we go down. We're going to focus today on the Shetland sheepdog, and I should say this adorable picture are 3 Shetland sheepdogs that are owned by Dana Drager, one of the postdocs in the lab, or now staff scientist who's worked on this. Okay, let's go back to our wheel. So this wheel is not made up of 170,000 random positions. It's actually made up of whole genome sequence. And represented here are 347 different dog breeds and populations. And I've simplified this somewhat. And again, divides up into 23 clades or populations. This is rooted in the, I think this one was rooted in the jackal, might have been the wolf. Out of this, we have 26 million single nucleotide polymorphisms, so tremendous power to do mapping. Again, it's a neighbor joining cladogram. And now when we impose on this, the dog breeds that you just saw on the previous slide that have the highest risk. All right, so we can use this wheel to figure out, well, can we combine any of these breeds in order to increase our power? Well, maybe we could combine the Scottish Terrier, and you can't see it here, but West Highland White Terrier would fall here. Maybe we could combine Samoyed, Keeshond, and the American Eskimo dog. But the Beagle, we would not be able to. That's going to be there by itself. And it's blocked out down here, but the same would be true of the Shetland sheepdog. So our interest is in the Shetland sheepdog, so we're going to have to focus on that by itself. Okay, not sure why that happened. All right, so we did a genome-wide association study in the Shetland sheepdog, and as you'll see in a moment, in related breeds, and that identifies a region on canine chromosome 13. It's a pretty big region, but there are 3 adjacent single nucleotide polymorphisms that really highlight that region and are statistically significant, with a p-value of 3 × 10⁻⁷. And that's after correction for population structure. It remains significant after 10,000 permutations. You're looking at a standard Manhattan plot going from the top of chromosome 1 all the way to the bottom of chromosome 38 and the X chromosome. And you can see the line of Bonferroni correction and the very strong signal that we get here. Now notice we have 49 cases and 50 controls. And when you set these up in dogs, it's not the number of samples that matters, it's actually the number of lineages that matter. So we design our studies so no cases, for instance, share common grandparents and ideally even great-grandparents. The same thing with controls. So we're sampling the maximum amount of diversity that exists in the breed. And so we're really sampling many more than just those 49 cases and those 50 controls. Now, if we work within one breed, as we did in the previous slide, lots of Shetland sheepdogs, and we assume the mutation occurred, in some dog ancestrally, canine chromosome 13, then we can look at the meiotic recombination that's gone on between the green and the gray chromosomes over time, so that each dog has slightly different recombination events. And that means when we think about the region, we have to look for our gene and mutation, and it's pretty big. In this case, it's going to be for sure at least a third of the chromosome. But now we make use of that wheel and we combine data from related breeds, the Collie, the Australian Shepherd, the Border Collie, the Kelpie, the Cardigan Welsh Corgi, all dogs that were in that same clade. Much more meiotic recombination has occurred over time as those breeds have been developed. And now the region that we have to look at to find our mutation becomes much, much smaller, and we have a great deal more significant. So here is our original data set, canine chromosome 13. It was somewhere in this 1.5 to 2 megabase region. But now we're going to combine data, if you look on the right, from the Collie, the Shetland sheepdog we started with, the Australian Shepherd, as well as the Border Collie. So now we go from a p-value of 3 times 10 to the -7 up to 5 times 10 to the -10th. And if you look at the bottom of the slide, again, this is work that's been led by Heidi Parker, obviously in collaboration with Debbie Knapp in her lab and others in my own lab. And the signal is much stronger and it's much narrower. So we have a much better idea where we want to go to try and find this gene. So originally this was a pretty big region, 2 million base pairs. There were about 20 genes in this region, and those genes are listed for you down here. We had whole genome sequence from 7 cases and 7 controls that were Shetland sheepdogs. And when we looked at that, we found about 2,000 potential variants within this region that followed the allele pattern of those most associated SNPs, or single nucleotide polymorphisms, from the original GWAS. Now, as you'll see from Dog 10K, we have thousands of other dogs whose genomes have been sequenced. So we looked at those 2,000 in 1,700 whole genome sequences from lots of other dogs of lots of other breeds. And that took us down to about 253 variants that were in predicted regulatory regions when we aligned the dog over the human genome sequence, as well as 20 variants that are within genes. So just to talk about those first, they led us to the gene Nepal1. So we found a coding variant with a minor allele frequency of less than 0.001 when we looked outside the clade in 1,700 whole genome sequences. It causes a glycine to aspartic acid within the 6' transmembrane domain, which we know is functionally very important. We applied lots of statistical tests, and they were all very robustly telling us this is functionally relevant. Mutt-Fred predicts that this altered transmembrane domain protein is functionally relevant. It has a strong pathogenicity score with a low false positivity rate. We looked with VEST, and we got a p-value of 0.003, and CHASM, the canine driver impact score, 0.02. Now, I will say we don't yet know it's this base pair, although certainly that's where our strongest evidence is, but I would be negligent if I didn't tell you we also have a putative enhancer variant very close by. It's an enhancer for Nepal1, and it's in near perfect linkage disequilibrium with the mutation. So the mutation is starred here in Canis familiaris, but you can see from the red coloring that this is a very, very highly conserved region, going all the way from mice down to frogs, for instance, and certainly everything in between. So we know we're probably on the right gene and pretty close to the right region. Now, Nepal-1 is very interesting. It's overexpressed in canine bladder cancer, and that's been shown in at least 3 independent studies. It's also overexpressed in canine bladder cancer cell lines. Increased expression is associated with poor survival and metastases in a human cancer, human oral squamous cell carcinoma. Now, the gene is involved in ion transport, primarily but not exclusively magnesium. And this mutation is expected to alter the ability of the gene to transport magnesium and maybe even other ions across cellular membranes. This overall pathway is crucial for tumor development, and it's dysregulated in both dog and human bladder cancers. So this is part of a larger family of ion transport channels, which we know are very important in lots of different cancer cell types, either overexpressed or mutated. And that's a completely topic for maybe another day to talk about all of those cancer types. The association peak is interesting because it actually coincides with a region of overexpressed genes when we look at tumor. So again, this is the second time I'll talk about tumor. So here's our original peak on chromosome 13. And now if you look at that second panel, okay, blue, we've blown that region up. Blue is the GWAS, or genome-wide association study. And then black is the upregulated gene cluster. So you can see a peak here in blue in this big blow up. And to the left, you see a second peak, which is this upregulated gene cluster. So is it the exact same base pair that's relevant in both of these? No, I don't think so. That wouldn't make any sense. But it may be a cluster of genes that's relevant. And in fact, the mutation that occurs in the enhancer is known to affect regulation of 10 genes in the region, including, of course, Nepal-1. So it's not a completely simple story. There's multiple things going on, but we now know the very tiny region that they're going on in, because we could take advantage of all the information we had about different dog breeds and from the thousands and thousands and thousands of DNA samples we have in our freezer. Now, we also know that there's other loci that are responsible, that are contributing to the disease. And so Heidi did 2 more genome-wide association studies. She did one where she controlled for Nepal-1 using all homozygous cases and controls. They all carried the mutation. And then one where they were all heterozygous. So one allele carried the mutation and one did not. And now other loci are popping up on canine chromosome 28, on canine chromosome 9, as well as on canine chromosome 4. We would have expected this. No cancer is a single gene story. Even those examples like BRCA1, breast cancer susceptibility, there's still going to be multiple genes that play a role in agent onset, in expression, in response to treatment, in presentation, in location. So it's interesting here. We've spent a lot of time tracking down the other haplotypes associated with some of these other genes, the one on chromosome 28, as well as those on chromosomes 4, 9, and 21. So you can actually build a diagram that looks like this. And so you can start with the original G to A mutation, and you can be a homozygote for one or the other allele, or you can be a heterozygote. And then you can work your way down and add in, well, what happens if you have the minor allele at chromosome 21 or chromosome 28? Over here, what happens if you have the major allele at chromosome 21? And you can consider all of these, as well as the alleles on chromosome 4 and chromosome 9 and chromosome 1. And what it comes down to is that certain combinations will increase your risk of disease, whereas other combinations will decrease your risk. For sure, the mutation in NEPAL1 increases your risk, but it can be ameliorated by these other combinations of alleles such that you end up with a decrease risk, or you can end up with no change at all, depending on how your patterns of yes and no's go. So that's what cancer does look like, right? We know it's never a single gene story. And it's really a combination of heterozygote and homozygous states that end up putting you where you are in terms of your cancer risk. So our hypothesis is that the NEPAL1 mutation increases risk certainly in the homozygous state. Risk is also increased by the disease-associated variant on canine chromosome 28. That's a biggie. And then these others are more minor, 4, 9, 21, increase or decrease risk depending on the allele combinations. And so we continue to work on this, we continue to refine this, and we continue to work down to identifying the functional mutations. Now, people always say you give these seminars on purebred dogs. Well, what about mixed breed dogs? Well, mixed breed dogs are increasingly a big part of the story. So you may know that dog variants are tracked in something called Online Mendelian Inheritance of Mammals. It's a free resource that's kept or developed by investigators in Sydney School of Veterinary Medicine. It's a compendium of inherited disorders, other single locus traits, associated genes, and variants in 538 animal species. There are 152 at the time I made this slide disease-causing gene variants that were in the catalog for dogs, and most were originally described in purebred dogs. Now, in 2018, a report was published on 100,000 dogs to see how many of these 152 variants could be found. 80 of them were seen in 80 of them were seen in purebred dogs and in mixed breed dogs. So of these, 53% were seen in purebred dogs and mixed breed dogs, but there were 34 of the 152, or 22%, that were only seen in mixed breed dogs. And that was surprising because it's telling us that mixed breed dogs are a reservoir for disease mutations that have essentially been eliminated or somehow acquired additional mutations in purebred dogs. So now dogs like Sophie, who's 73% Collie, Bogey, who's 27% Australian Shepherd, and Grace, who's 12% Collie, these are all at-risk breeds for our bladder cancer. And suddenly they become very useful because they have very small parts of their genome that are in that at-risk category. And I should say, we found 3 mixed breed dogs that were part Shetland sheepdog, all of which carried the exact same NEPAL1 mutation we found in the Shelties. So these mixed breed dogs, depending on their component breeds, end up being extremely useful. Okay, so why are we able to do all of this? Well, in just these last few slides, I want to say a few things about Dog 10K. So Dog 10K is an international effort that I, together with Bob Wayne and Guodong Yang, started in 2019 to generate and analyze 20x whole genome sequence. In the end, we're hoping to do 10,000 canids. We wanted to capture the genetic diversity underlying the phenotypic and the geographic variability of living canids. And this included breed dogs for sure, which we're talking about here, but it also included village dogs, certain niche populations like populations of dogs that aren't really breeds but live in one geographic region, as well as wolves and coyotes. And we wanted to develop this resource and make it publicly available even prior to publication, which we did, to allow the wider community to ask questions regarding domestication, breed formation, aging, behavior, morphology, and health. All of the samples were sequenced in the same way at the same place. They were all processed in a harmonized fashion to reduce batch effects. The processing pipelines and the data have all been released so that the community can take your own data of a bunch of dogs you've sequenced and you can add it to the larger data set. So just give you a quick overview. These are all samples that were newly sequenced. They hadn't been sequenced before. As expected, half are male, half are female. There are 194 breeds or populations that are unique to this panel. So we greatly expanded the number of breeds that we had genetic or genomic information on. We asked people to contribute samples that were disease-free, and I know that might be frustrating, but we wanted this to be a reference data set. So at the time that they were provided to us, the samples were disease-free, and we got really close to that 20x coverage. And there are at least a minimum of 3 dogs for each of those breeds, and obviously many more for most of them. The data in the pipelines are available. We only align to CanFam 4, which Cheryl distinguished for you from CanFam 3. So this is the German Shepherd, Tasha, that was sequenced at the Broad. Tasha's female, so we did include the Y chromosome. It had been provided by Jeff Schonenbeck at Edinburgh from Labrador Retriever. From the same individual, we got single nucleotide variants as well as structural variants from all the chromosomes as well as from the X. We looked at the mitochondrial genome, and of course, many times we can't get 20x. It's expensive, or we don't have enough DNA or enough good quality DNA, and we can only get 1x or 2x. And so we did develop an imputation reference panel that works just exceptionally well. So if you look on the right side of the slide, you can see that we had 1,600 breed dogs, 18 mixed and other dogs, 336 village dogs, about 68 wolves and 4 coyotes. And then we took this through a series of filters, initial QC. We then went through a very strict filter. We cut the data set even more when we did the mitochondria work, and we cut it the most for structural variants because that's, of course, the most error prone. And then you can see all the different things that are in the paper, which is published in Genome Biology at the end of last year. We looked at SNV sharing, breed relationship, runs of homozygosity, selection, and imputation. So basically demography and genome architecture. We studied function, and again, more genome architecture, looking at functional annotation, genotype concordance. We coordinated this with the OMIA data set I talked about on the previous slide. We looked at constraint, and we also looked at metabolism and drug targets. For the mitochondrial analysis, we looked at haplotypes and geographic distribution, the latter being particularly interesting to those studying domestication, and structural variation. Functional annotation, we looked at retrogenes, and again, metabolism and drug targets. So there's a vast amount of data in this paper, and a huge amount of it is in the massive number of supplements that are in there. Let me just give you a couple highlights. There were 48 million variants that we found. 12 million were discovered for the first time in this exercise, unique to Dog 10K. 34 million of those are SNVs, 14 million are indels of 50 base pairs or greater. And then we had 140,000 structural variants greater than 50 base pairs, including vitagenomes. If you look in this second panel, you'll see that the distribution of SNVs is not even, and you're looking up and down each one of the chromosomes. The PAR region on the X is indicated for you. It's not even, but we certainly didn't expect it to be. Over here in the first panel, you're looking at mixed breed dogs, village dogs, and wolf. And you can see a tremendous amount of overlap, but you can also see regions of non-overlap. For 22 breeds, the vast majority of variation has now been found. There's really no point in continuing to sequence those. Most things are in the middle, but interestingly, there are about 20 breeds for which, for 22 breeds, there's no point in continuing to sequence. The vast majority of variants have been identified. And for about 75%, for another 20 breeds, less than 75% of total variation has been discovered. So those are odd breeds. And certainly if you're interested in those odd breeds, they're in the paper, and more time could be spent on those. And most things, of course, are in the middle. And then finally, the variants are very useful for distinguishing populations. And so you're looking at a PCA analysis, a principal components analysis, and you can see the color coding distinguishes wolves, wolf dog hybrids the Western European breeds, which is what, of course, most of the breeds in the United States are, Western village dogs, Eastern breeds, and the Eastern village dogs. And about 10% of the genome is under constraint. So there's strong selection for there not to be change there. And when there is change there, that's when we tend to see disease. And my lab is doing a much more expansive study on that right now. Interestingly, the constrained bases are in the same general place as they are in humans, but as expected, they're not always in the same position. So this is my last slide. And this is a neighbor joining tree now based on sequence. It shows you haplotype sharing for 332 breeds, clustered by type, geography, appearance, occupation. And here are groups, and here are subgroups, and here are historic occupations. And I'll just leave that up for just a second. I mean, this is a figure that, again, Heidi Parker made. Just an extraordinary amount of work to pull all of this together. Let me just tell you who's done the work. The TCC study that I talked about, pretty small study. In my own lab, it was led by Heidi Parker, and in Debbie's lab by Deepika Dewan. Many other people in our labs contributed in indirect ways who are not listed here for you on this slide. This is Dog 10K, and there's just an extraordinary number of people, all who donated their time to this study. We did not have a great big unifying grant to do this. This was really the motivation of people in the community, and they're going to put the QR code up for you so that you can go ahead and access everything that you would ever want. And, you know, it's all going to be in those supplements. So I'll go ahead and stop there. Thank you.
Thank you so much. That was incredible. I am really, really pleased that we did this symposium. And I want everybody here to know that next month we're having another 2 speakers on canine cancer topics. Details for that will be posted to the website shortly. We have a lot of questions in the chat, so I'm going to start at the beginning and voice them. Some of them have been partially answered, and I'll also mention those answers. But hopefully our speakers are okay to hang out for a few more minutes.
Yeah.
Great. Fantastic. So I'm going to start with a question about the Integrated Canine Data Commons for Gina. Was any work done to map ICDC studies information, diagnosis, pathology, morphologies, body structures, and others to a common terminology like SNOMED CT?
So in the current phase of the project, within the new scope of work, we are looking into incorporating common data elements from the CADSR. So up until this point, there's not been any mapping that's been formally done, unless it was maybe more at the study specific level within those individual groups, not on the ICDC level. However, for the data that we have and for our data model, we are looking into incorporating common data elements.
Good, cool. And then we have a question for Dr. London. Can you tell a bit more about the probe set for the 10X platform? For example, what kind of panels are you designing, and can you say something about the vendor?
It is a whole tumor.
Okay.
So it's basically all genes. We're mapping the entire whatever we can. So it's not a limited panel the way it was with nanostring. It'll be everything. So it should be a whole tumor atlas, which is going to give us a lot of information, which is why it's going to be very valuable. Yeah.
Amazing. So we also had a question that spawned some vibrant commentary. I'm going to read the question and the comments. Based on the genetic and histological similarity between humans and canines, it makes perfect sense to study canines. However, I can't help but wonder, what are the limitations to using canines as a model organism as opposed to something like mice? And we had a response in the chat. Canines are human-owned, so there is more of a clinical trial component, but much less difficult or restrictive than human clinical trials. You may have to limit repeated sampling. There's difficulty getting owners to go back to the clinic repeatedly, or keep to minimal invasive methods if what you need is outside established treatment protocols. For example, people don't want their pets to undergo unnecessary surgery for the sake of being a control, so mock surgeries are out. Mice have their own limitations. Mice don't naturally get the disease, and current mouse models have their own limitations in regards to relevance to human disease process. We've cured cancer in mice many times, but that hasn't always been relevant to humans. It probably boils down to your goals. Elaine and Cheryl, and Gina,
Sure.
do you have any?
Yeah, I would just
follow-up?
like to jump on this because, like, I think having been in this field for a really long time doing translational work, I think the point I would like to make is there is no one perfect model for human cancer, and even human cancers aren't the best model for human cancers. So I think the really cool thing about biology is that stuff that's important tends to be conserved, and you can find threads of that sort of conserved biology if you use multiple model systems. So the goal isn't to say we should be only using dogs, it's to say we should add more models to the system so that we get different types of data, and then can identify the signal and the noise, because that actually becomes easier to do when you're using different approaches in different model systems. No one model is ideal. This just adds value in a different way.
And I'll just add to that by saying, when you're looking at the genome, you're looking at the germline. There's tremendous advantages in terms of power and breed structure, and spending a lot less time and money to get down to small regions of the genome where you think your mutation is. But we always go back and say, what's known about the human genome in this region, because it's just been so exquisitely well studied. What's known about the mouse? What's known about the cat? I mean, we look at a variety of different animal systems, and it's, as Cheryl said, it's comparative genomics, really. It's our own interpretation of One Health. But for us, for us studying the germline and susceptibility, this is a great place to start because of breed structure.
Thank you so much. I also wanted to highlight what our commenter said, and also what you had in the beginning, Dr. Ostrander, about the fact that dogs live with us. Dogs are exposed to the same environments and the same small scale environments that we're exposed to. And that is not something that I appreciated before this, and seems like a very valuable part. We had another question. Big thanks for making ICDC a single stop platform for most of canine data. Agreed. I also want to give kudos to that. Question: for working on the Seven Bridges platform, are there any egress fees for computing? So I think I'm actually going to just answer this myself as a community engagement manager for the Cancer Genomics Cloud. There are no egress fees to move data from ICDC to a Seven Bridges Cancer Genomics Cloud workspace, because this data is all being accessed through the Cancer Research Data Commons interoperability structure. So you can link to the data, you can access the data from ICDC in your Cancer Genomics Cloud workspace, and you aren't going to be paying for data egress or even for data storage, because that data is still actually stored on the ICDC or the Cancer Research Data Commons servers. There are charges for computing, but those charges are direct cost. We pass through the Amazon Web Services charges directly. And there are also compliments to the National Cancer Institute, $300 in cloud credits available, and also some other mechanisms available for allaying those costs. I will plug the CGC a little bit more at the end of this. So we had another question: how comparable is this database? You may have meant the ICDC to the TCGA. Specifically interested in read depth and availability of neighboring unaffected tissues, as well as blood for comparison. Also, can you download, transfer the raw FASTQ files to a server? And Gina, you had answered this in chat. So do you want to read your response?
Sorry. don't know where it
I've
is
got
there,
it.
but
I'll
okay,
read
you
it to
you
you.
can read it. Yeah.
Yeah, the
Thanks.
ICDC Best Practices Subcommittee is working to implement standards for read depth and tool filtering and parameterization. The raw FASTQ files for most studies in the ICDC are accessible through the NCBI Sequence Read Archive. We are considering adding FASTQ files in addition to the BAM files for each study to the ICDC so that they can be easily exported for analysis into the CGC. And I would just like to add to that, we also have very easy to use tools on the CGC that can take a BAM file and produce the FASTQ files. So yeah. As relates to how comparable is this database to the TCGA, I don't have a super good grasp on the ICDC's data that's available, but I know the TCGA is enormous. The TCGA is 2.5 plus petabytes.
Right. We're not quite there. We're moving in that direction. So we're coming in hot at 3.23 terabytes. And as far as how it compares directly with TCGA, at least in the context of read depth, I'm not exactly sure because for each of the studies within the ICDC, the data has been generated by all different groups. And I would assume, at least from the data that that I've personally worked with from the ICDC, the read depth is different from study to study.
And then I'll also say in work that I've done with ICDC data, I have seen that there is lots of whole blood normal samples available for affected patients. I have not seen very much neighboring normal tissue, but we just heard from Dr. London that with the advent of these new panels, we're going to be able to start looking at formalin fixed tissue. So
I'm not sure if Dr. Knapp is, I'm not sure if Dr. Knapp is still on the call, but she may be able to speak to, we're looking to add additional normals. And if she's still here, she might be able to speak to that.
I'm here, but I don't have any updates right now.
Okay, sorry to put you on the
Working on
spot,
it.
Debbie.
No problem.
Okay, we're
Gina-
waiting on more normals, so I know they're coming at some point.
that's
Gina,
awesome.
Is read depth something that we could incorporate into the ICDC like parameters so that way you can see before you select your cases? Is that something that we could incorporate forward or?
I think that's a good idea. I think we are looking to incorporate, at least through the user interface, some additional metadata related to the actual files as far as like bioinformatic information. So right now that information is accessible if you go into JBrowse, which is a next generation genome browser. So if you have a BAM file in the ICDC, it's very easy. We have integrated JBrowse into our application. So you could pop that sample open within JBrowse, and now you can check out, inspect directly the read depth and other metrics associated with the sequencing. But I like your idea too. So I will definitely bring that up for the backlog discussions.
Yeah, awesome. Thank you.
We have another question that I think also speaks to this, to the hunger for more canine cancer data. Are there plans for a canine telomere to telomere genome? And Dr. Ostrander just
answered
responded.
it. Yeah. So Jeff Schonenbeck is really the person who started to motivate that, and he's working with Adam Philippi here at NHGRI. And most definitely there is, as well as a pan genome. So yes, we all see the value of a T to T.
We also have some discussion in chat about the utility of CanFam4. I was also fully convinced that we have to be hosting CanFam4 on the CGC and in this workshop. There's one question
About the mitogenome. They're
about
right.
the mitogenome.
Yeah. So I believe there are plans to annotate it for sure, because there's so much interest in from the ancient DNA crowd. And so you may not be aware, but there are people throughout the world who are doing amazing sampling in just exotic places and able to get DNA. Some of that DNA has been sent to NIH for sequencing, They've made libraries, we've done sequencing, and they've just been really great partnerships for learning about the evolution of alleles and variants and migration of human peoples. And so, yes, for sure, there'll be annotation of that. And there are a number of reference genomes, and we do go to others of them for various things. If we find something in CanFam4 that looks great, we then go look at the Great Dane. We go look at the revised version of Tasha. You know, we might go look at the Senji. So please feel free to make use of those other reference genomes as well. We start with Tasha, the German Shepherd, and we move out from there.
Awesome. So one other question that we have from someone who is preparing to do some work. Dr. London, how is your experience in doing the NanoStrain Canine Cancer Atlas on osteosarcoma? As someone who is also planning on doing this, was it difficult to pre-treat the tissues in order to get adequate RNA probe binding? And was the bone adequately cellular? Tumor is, but did you also have bone E-sample? Most importantly, any plans to publish that data anytime soon?
Thank you for that question. So we spent a lot of time optimizing the preparation of those specimens. So we use Cal4 as a fixative, which has helped tremendously with the decal and stabilizes the RNA. So we went through a whole process of optimizing it so that the fixed tissues were decaled and had good RNA quality. We used RNAscope to just verify that we have good RNA quality as prior to doing the transcriptional, the transcriptomics on the genomics platform. The largest challenge I think we've had isn't so much with RNA quality, it's just optimizing antibodies and choosing regions of interest. The ROI selection can be difficult. In some cancers like osteosarcoma, there's just not a huge immune infiltrate, and so there's really few T cells. And other cancers, it's easier to pick out those ROIs. So ultimately, at the end of the day, the largest barrier is essentially getting your antibodies optimized on the platform so you can filter and choose your ROIs. We're working through the data sets, a lot of data. It also just generates an enormous amount of data. And so we've had to build some of our own pipelines for analysis and get things, again, lined up to CanFam4, which took some time. So it's coming. It just takes time.
Awesome. Thank you so much.
You're welcome.
So with that, we are 20 minutes past the hour. So I'm going to go ahead and end things here. I want to remind people we will be picking up this discussion again next month. And we will be sending details out about that shortly. Thank you so much to everyone who attended.
Thank you.
And let's all thank our speakers.
Thank you, everybody.
Thanks,
Thank you.
everybody.
Thank you.
Thank
Bye.
you.